So welcome to 4Keys Private Tuition, 4Keys College of Science Education. I'm your host, Seshungo. So we'll proceed from where we left in the previous video. We're talking about the advantages of biodiversity. We're talking about ecosystems, pyramids of biomass, etc. So let's proceed to the next section, which is plant nutrition. And we'll talk about photosynthesis. Photosynthesis, leaf structure. You're supposed to know these things. So let's talk firstly about the internal structure. That would be an external structure. You see a lot of veins there that carry for you water to the leaf. Let's talk about the internal structure. So there's another diagram that I shall present later on, which shows you these layers that we'll talk about here. Firstly, the cuticle, which is made up of wax, a waterproofing material. And we have an epidermis, which is thin and transparent to allow light to pass through. We also have all those layers: palisade layer, spongy layer, vascular bundle, and we also have stomata pores at the bottom of the leaf. So we looked at specialized cells earlier on where we met these palisade cells. So remember, they are responsible for carrying out photosynthesis. So there's a region which is called the palisade layer in the leaf, which is the main region for photosynthesis. It is packed with these palisade cells that contain, remember, they contain what? Many chloroplasts, which contain chlorophyll to enable to capture sunlight for the process of photosynthesis. We have spongy layer, which contains spongy cells that are spaced, and this allows for the easy diffusion of gases through this layer towards the palisade layer. So we have carbon dioxide, which diffuses into the leaf because it is needed for the process of photosynthesis. We have a vascular bundle, which is made up of xylem and phloem vessels. Remember, your xylem vessels carry minerals and water, and phloem vessels will carry for you the sugars that are produced during photosynthesis. We have stomata pores that are found on the underside of the leaf. So I've presented this in another video where we were talking about an experiment that is done to show that a lot of stomata are found on the underside of the leaf compared to the upper side of the leaf. So it's important for you to check that video as well. So remember, stomata pores are open and closed in response to the light intensity. If there's a high amount of light, then the stomata pores will open to allow carbon dioxide to enter in preparation for photosynthesis to take place. They have guard cells which control this opening and closing. So this is also related to the process of transpiration, which is the loss of water vapor through the leaves of a plant. So a lot of water vapor is lost through the stomata pores when there's high light intensity. So light intensity happens to be a factor that affects transpiration. We'll look at that again later on, but you can draw the connections here. So the stomata pores will open when photosynthesis is about to take place, and these allow for gaseous exchange where we have our carbon dioxide entering the leaves. Okay. What are the adaptations of leaves for the process of photosynthesis? Most leaves are broad. This offers a large surface area for the absorption of sunlight and also absorption of carbon dioxide. Most leaves are thin, and this allows for easy diffusion of carbon dioxide so that it reaches all the cells, and also for easy penetration of sunlight so that it is able to reach all the inner cells. They have many chloroplasts. As we saw, these chloroplasts contain chlorophyll, which traps sunlight for the process of photosynthesis to take place. And we have spongy cells that allow for easy diffusion of carbon dioxide inside the leaf. Remember, those spongy cells are spaced to allow for the easy diffusion. Large spaces between the spongy cells, as we are saying. Then what is another adaptation? There are many stomata pores for the exchange of carbon dioxide and oxygen with the air outside. So remember, photosynthesis also produces oxygen, so there is a gaseous exchange that takes place. Carbon dioxide enters whilst oxygen is leaving these cells. And we have palisade cells that are closely packed and elongated to increase the surface area for the absorption of what? Sunlight. We have a branching network of veins which provides a good water and mineral ion supply, okay, from the roots. So we have factors that affect photosynthesis: light intensity, carbon dioxide concentration, water, chlorophyll, and temperature. So you must know that these are the factors that affect our process of photosynthesis. How do you test a leaf for starch? If you want to show that photosynthesis was taking place, then you have to test a leaf for starch. If starch is present, it shows that the leaf was carrying out photosynthesis. If there is no starch, then the leaf was not able to carry out photosynthesis. So testing for starch is important. The first stage is to boil the leaf in water in order to stop all the biochemical reactions, or simply to kill the leaf. Stage number 2 is to boil the water in a water bath inside alcohol, and this is to remove all the chlorophyll. Stage number 3 is to put the leaf in warm water to soften it, and lastly we have we put our leaf on a white tile. We add iodine solution. Iodine solution is brown, but when there is starch inside the leaf, it would change to blue-black. So a blue-black color is a positive test for starch. You must remember that. So those are the stages. You must try to remember the reason for each stage. And there are precautions that you must be aware of when you're carrying out this experiment, which is to extinguish all the flames, because alcohol is highly flammable. So that's an important precaution there. And also to boil the leaf in a water bath instead of heating directly onto the heating tube that contains your alcohol. Again, for the same reason that your alcohol is highly flammable. Test for oxygen. How do you test? Use a glowing splint. Oxygen will relight a glowing splint. There's an experiment that can be done to collect oxygen from photosynthesis, as shown there, and you can put a glowing splint there. When you remove your inverted test tube there, you can test for oxygen, and the oxygen is supposed to relight a glowing splint. So this experiment can be done to collect oxygen, again showing that oxygen is produced during photosynthesis, and the oxygen can be used, can be tested using a glowing splint, wooden splint. What happens to the end products of photosynthesis? What are the products of photosynthesis? We have our glucose, or carbohydrates, and oxygen. So what happens to the glucose? It is converted to starch. So remember, it is the leaves will produce glucose, which is then converted to starch. Starch is a good storage molecule because it is insoluble in water. So it can be stored in those storage organs, or your tubers, for example. This is where we store the starch. It can be stored in the leaves themselves, in the stem. So this is important. The glucose has to be converted to the starch, the storage organ. So there's also another process which is called translocation, where if now the plant wants to use the starch, it is converted back from the starch back to glucose, or sucrose, another soluble simple sugar, and it is transported through the phloem vessels in a process that is called translocation. So remember, translocation, the transport of simple sugars through our phloem vessels. Okay, so some of the carbohydrates are also used. They are converted to amino acids, which are used to make proteins and enzymes for the plant. They are converted to cellular cell wall formation and lipid formation. So you can see there are a lot of uses for the glucose. The oxygen simply diffuses out of the leaf into the atmosphere. So let's end here for now. We'll meet again in the next video, where we'll continue with our study of biology, combined science. You can always contact me for further explanations. We'll do actual exam coaching, where I'll share with you statistics. I have been compiling statistics for past exam. All the exams that I delivered, I've been compiling statistics. Ma statistics anoshowa ma questions anonyanya kufailiwa, ma questions akaomera vanhu. I have all that information, and now it's available if you'd want further help with combined science. So let's meet in the next video. Enjoy your study.